almost all amino acids have their own transaminases. Almost all amino acids have their own transaminases that catalyzes this transamination reaction. And there are three features of transamination reaction. One is all transamination reactions are reversible. And then second feature is transamination reaction. You will see glutamate and alpha ketoglutarate as reactant, or one of the reactant and one of the product. If this is reactant, this will be product. This is reactant, this will be product, because this is reversible reaction. Now third point, it will always have PLP. So vitamin B6 always be there. You can see these 3 points. These are reversible. All are reversible reactions. All are reversible reactions. All transamination reactions are reversible. They will have one of the, here you can see alpha ketoglutarate here, alpha ketoglutarate as reactant. One of the reactant is alpha ketoglutarate. And here, if you see the reaction this way, you can see, if you see this way, this is a product. But if you see this way, this will become product and this is reactant. So reactant and product will always be, one of the reactant and product will always be alpha ketoglutarate and glutamate. So remember this is this 3 point, and third point is this pyridoxal phosphate. One is reversible, second is this, and third is pyridoxal phosphate. This is transamination. Now transamination, as you know, how do you name the enzyme? You write the name of substrate, and then you write the transaminase function. So you write the substrate name and then function, and you put age. So you see glutamate, pyruvate, amino trans. So alanine amino transferase, because alanine gives amino group to this. So here vitamin B6. What does this do? From alanine, this amino group will be taken by vitamin B6. This pyridoxal phosphate is vitamin B6. And then this vitamin B6 transfer this amino group here. So this is how transamination. So this is transamination is mediated by vitamin B6. This is the role of vitamin B6. Now this alanine becomes pyruvate, because keto acid of alanine is, this is alanine. You can see alanine will have amino group here and acid group here, and then amino acid, and this is CH3 group, and this is alanine, and you can see pyruvate. This is pyruvate. Pyruvate will have this type of group, CH3. So I am writing here pyruvate, here alanine. Don't confuse with that. So you can see this is pyruvate. Isn't this pyruvate? So pyruvate, this is replaced by amino group. This is alanine. So you can see this amino group, when you remove, this become pyruvate. So this is what removal of this amino acid is becoming pyruvate. So alanine converting to pyruvate. Here aspartate. Aspartate is converting into, now aspartate is converting into oxaloacetate, because keto acid of aspartate is oxaloacetate. Aspartate is a 4-carbon compound, 4-carbon amino acid. So you can see this is 4-carbon amino acid. This is acid here, and then amino group will be here. Now this is acid, amino acid, and double carboxylic amino acid, 2 carboxy group containing amino acid. So this is aspartate. When you remove this NH2 and give it back to this will become glutamate, and now aspartate will become like this. It will become like this. This is oxaloacetate. So keto acid of aspartate is oxaloacetate. For valine, keto acid of the same way, if you draw, I'm not going to draw the structure of valine. So valine will convert into isovalerate. You can easily remember. A valine will convert into isovalerate. Let me draw the structure of valine here. Here I will draw. Okay. So this is acid, and then what? Amino group, amino acid. So valine will be this. So you add a V type of structure. So see here V, CH3 here. Yes. So this is a valine. Now valine, when it converts into this, so this will become CH2, and then CH3, and then CH3. So this is 5, 1, 2, 3, 4, 5. So valeric acid, 5 means, valeric means 5. So you can see V and 5. V is Roman 5. So you can remember that valeric is 5. So if there is 5 carbon, it is called as valeric. But branch chain is there, that is why isovaleric. So keto isovaleric. This is a keto-isovaleric. So valine will convert into keto-isovaleric acid. So this is the reaction you can see. So did you understand? One amino acid is being converted into keto acid, and then this keto acid being converted into amino acid. That why does it happen? Because of transamination. Amine group is transferring from this one amino acid to other keto acid. Here, one amino acid to keto acid. Amino group is being transferred, and transfer happens through pyridoxal phosphate. And that is why this aspartate becomes oxaloacetate and this alpha ketoglutarate becomes glutamate. Did you all understand?
Yes, sir, but I have a question. Okay, so you mentioned that the reactant, glutamate always has to be a reactant, but like can't a reaction like alanine plus oxaloacetate yield pyruvate and aspartate without alpha ketoglutarate?
No. It cannot happen. That is why I told you. There will always be this, either of this as reactant. One as reactant, other will be product, always.
Okay, thank you, sir.
Okay, did you get that? There cannot be oxaloacetate. This will always be this or this. Can be reactant, and one will be reactant, other will be product. Okay?
Okay, thank you.
Okay, and you can just, you remember this enzyme name, ALT. This is very important enzyme. This enzyme is also used as liver function test. AST. This is AST. This is used as liver function test. So these are very important. So that means I'm telling you the enzymes that are utilized in liver function test also. So remember, these are used as liver function tests. This is very important enzyme. So you don't know, this is just a picture that's showing is like this. How does this pyridoxal phosphate will take this amino group? First, there it is inside the enzyme. It is tightly bound to enzyme, but it will take this amino group like this. A skip base formation will convert into taking intermediate skip base formation will form. Skip base means this type of with nitrogen carbon will make double bond, and one of the nitrogen bond with other carbon. So this is skip base. Both side carbon, in between there is nitrogen. So this is skip base. You can see here one carbon, one double bond in between nitrogen, double bond. So this is skip base. For intermediate skip base formation will take away this nitrogen here, amine, and then it gives amine to other. That is what I want to say. This is pyridoxal phosphate, so that you will remember the structure of pyridoxal phosphate. Pyridoxal phosphate is like ring structure. Okay, so this is vitamin B6 function. So intermediate pyridoxal phosphate will form. So you can remember this mnemonics for the function of, there are 6 function of vitamin B6. B6, 6 function. What is? First is, he is a very good man. He is heme synthesis. I have told you the first enzyme in heme synthesis, if you can remember, heme synthesis. If he is vitamin synthesis, we will talk about vitamin synthesis, vitamin B3 synthesis. And then G for glycogen degradation. We will talk about glycogenolysis, glycogenolysis. And then M for myelin synthesis. Do you remember sphingomyelin synthesis require vitamin B6? Myelin synthesis. A for amino acid catabolism. You have already seen this is amino acid catabolism. Amino acid catabolism. Not only this reaction, other reaction of amino acid catabolism also require vitamin B6 catabolism. And then N for neurotransmitter synthesis. This is a very important vitamin. So remember all these 6 functions at one place. He is a very good man. Just remember this mnemonics, you will remember. Now, all, as I have told you, all amino acids have their own amino transferase, transaminase. Okay, amino transferase. But there are few amino acids which does not undergo transamination reaction. These are this hydroxyl group, both hydroxyl group containing amino acid, threonine and serine. And one is histidine, other is proline. So 4 amino acid does not undergo transamination reaction. They will rather go deamination. Which type of deamination? Non-oxidative. Previously I told you oxidative deamination. Okay, so oxidative deamination of this glutamate will happen in liver. But this 4 amino acid will undergo direct. It will not undergo transamination reaction. It will direct undergo non-oxidative deamination. Non-oxidative deamination. That means it does not So hydrogen is not released. NADH is not released here. NADH is not released. Non-oxidative means NADH is not released here. That is what you have to remember. So this 4 amino acid undergo non-oxidative deamination. Okay.